Welcome. Welcome to today's webinar where Dr. Hamza will be presenting his latest work on machine and deep learning methods for enhanced MRI analysis of developing human brain. Our speaker today, Dr. Hamza Kebiri, received a bachelor and a master in communication systems engineering from École Polytechnique Fédérale de Lausanne and a master's degree in cognitive science, neuroinformatics, neuroscience from Osnabrück University. He worked in the Max Planck Institute for Brain Research in Germany, where he focused on deep learning segmentation and reconstruction of mice brain neurons imaged with electron microscope. After that, he performed a PhD thesis in the Faculty of Biology and Medicine of the University of Lausanne, and his thesis got the Best Thesis Award. During his PhD, he worked on reconstructing fetal and newborn brains from diffusion MRI with deep learning. He worked as a visiting scholar in Boston Children's Hospital, Harvard Medical School. He has also worked on various projects such as low-field MRI, ocular tumor segmentation, and multiple sclerosis lesion assessment. He is currently working as a postdoctoral researcher with the Center of Biomedical Imaging in EPFL and UNIM, focusing on various functional and diffusion imaging projects. His broad research interest encompasses neuroscience, consciousness, brain reconstruction, machine learning, and its application to brain imaging. During the talk, it will last for approximately 30 minutes, followed by a 10-minute Q&A. If you have any question during the talk, please feel free to ask them in the chat, and we will be sure to bring them up at the end of presentation. And without further ado, please, Dr. Hamza, the floor is yours.
Okay. Thank you very much, Iman and Erdwan and the whole MoroccoAI team for the invitation. So I'm very happy to be with you. So I will share my screen. So now you should be able to see my slides, right?
Yes, we can see.
Okay, yeah, so again, thank you very much. And big up for all the work you're doing and for the MoroccoAI community. Yeah, thank you for this, actually. So yeah, so I'm actually working in CIBM, and I'm in a transition period between my previous lab in UNIL and the next one in EPFL with the MIP lab. And this presentation will be basically around machine learning methods for applications in developing brains. And the idea is the presentation will be technical, but I will try to keep some level of abstractions and not go too much to the details so that we can have a larger audience. Okay, so I'll start with a brief introduction about why do we want to study these fetal and newborn brains. And then I will also talk about diffusion MRI and a bit about functional MRI. And then I will present one of the two studies that I worked on using deep learning basically for improving images or the models of diffusion images of newborns and fetal brains. And then I will present the last project of on functional MRI, basically using k-means clustering to split the neonatal thalamus into different nuclei. Okay, so why are we interested in the developing brains? So as you know, so the brain actually starts with the tube and then it's ends, let's say, as a completely folded organ. And most of the processes actually happen in the beginning of life. So there is actually a high speed of growth in the beginning, let's say between the 29 days to 6 months or 9 months, then maybe from 9 months to the adulthood. So this period is very crucial and studying it is very, very important. So there are many processes happening, such as cell differentiation. So there are like the cells are defined there, or like what is their job? Are they going to go to the cortex or to the white matter, or are they going to be subcortical, etc.? And then there is cell migration, so cell moves basically. And then there is neurogenesis, so there are new neurons that are generated, synaptogenesis, so new synapses, new connections between neurons. And then also synaptic pruning, so basically there are synapses that are removed during this period, and myelination and so many other important processes for the human brain. So one of the best methods actually to capture the brain in vivo and non-invasively is MRI. So MRI is typically so MRI has actually many contrasts. So depending on the physical process that you're probing, you can get many different information about the brain, and it's typically used with the T1 and T2 weighted properties, and these are somehow like inherent properties to each tissue. So, for example, gray matter has its own values, white matter has its own values depending on the age, on the disease, or etc. But here we are mostly interested in diffusion MRI and in functional MRI. So diffusion MRI, and I will briefly talk about it more. So it uses basically the water molecule displacements to get an information about the white matter. So white matter are basically these long myelinated axons that link the different brain regions, namely the cortex. And yeah, and then we'll also talk about functional MRI. So the last project was functional MRI, and basically functional MRI is just a measure about the oxygenated blood level in the different regions, and this is like an indirect measure for the brain activity. So basically, if you have a region that's fired in the brain, there is a rush of oxygenated blood that will go there, and this is what it's extracted there. Of course, there are other protocols, but this is not in our interest here. Okay, so basically, diffusion MRI relies on the diffusion of white matter of water molecules. So here we can see the ink diffusion in a glass of water, but actually, what we want to see in the brain is really the water molecules diffusing between the different cells and also inside the cells. And we want to use this information as a proxy to know about the anatomy. So basically, the idea is that if we try to extract the information along some directions, so for instance, here we will have a high diffusion of white matter, of the water molecule, sorry, in this direction, meaning that we will have less signal. And then in this opposite direction, we will have a restricted diffusion. So the water molecules will not diffuse enough. So we will have a different kind of signal between these two extreme cases. And of course, there are many other angles that we want to extract. So this is actually what's called the angular resolution here. So basically, we try to get the information about how the water molecules diffuses along different their directions. They're called gradient directions. And typically, you can acquire around 30, 60, or even 100 or more than 100 directions. And you can think about it as a labyrinth. So let's say your eyes are folded and then you are in a labyrinth and you cannot see. So the more directions you can actually go through, the more, let's say, you can know where you are and you can get a better estimation about the anatomy or the architecture of the labyrinth. So this is basically what we will tackle here. So this is a project that I started in Boston and then I continued here in Lausanne. So basically, the idea is to use deep learning to estimate these white matter fibers, but only using a small number of directions. So the goal here, so in this image here, what we see is basically it's a voxel. So a voxel is just a 3D pixel. And the goal is to estimate these fiber orientation distribution functions that are represented in each voxel here. And basically, they are represented by peaks. So here, for instance, we have two peaks. Here, let's say we have one peak. This is the corpus callosum. And basically, each peak, let's say, represents a bunch of white matter fibers. So the methods actually in adult brains require a lot of diffusion directions, and this is not possible for the developing brains because of the sensitivity of the cohort, namely the fetuses, for example, inside the womb of a mother, and also the newborn. So typically, we acquire around, let's say, 10, 20, or maximum 20-ish directions. But these methods usually need more than, let's say, 60, 50, hundreds even. So the goal here is basically to use deep learning and then really try to predict from these few measurements the target FOD that we want to predict here. So we basically use this architecture here. This is basically a unit like that for which we added many extensive connections. And the input to the network is basically the patches that go through like a spherical harmonic decomposition. Let's say just the patches here, and then each patch is put as an input, and then let's say the corresponding output FOD that is reconstructed with 300 directions from this research data set of the developing human connectome project is reconstructed. And then basically we train the network, and then the goal is that it really maps the small number of directions to these FODs. And actually it worked. So in the middle is this deep learning method, and on the left is the ground truth, so with 300 directions. And on the right is the typically used method, classical method. And we can see actually that the network quite learned, and it uses only 6 directions plus a B0, so 7 measurements, let's say. So we also compare our method with different classical methods and also deep learning methods. And we see that it either compares level on par with them, or even goes beyond them in some metrics. And even the deep learning ones. So for example, there's MLP here, or the city track that relies on transformers by a small margin, but still. And of course, when we increase the dimensionality of the input, so when we have more directions, we can see that the method actually performs better. But the good news is that it seems to plateau around 28 directions. So probably if you want to use a deep learning method to predict these FODs, we don't have to go, let's say, for more than 28 directions. The information is actually there in the learning. Okay, so here the idea is actually to also make the network work for fetal brains. So these are fetuses, images of fetuses from Boston Children's Hospital. And the trick here is that we trained basically the network on preterms. So preterm newborns are basically the newborns that are not born at term, so before 38 weeks in this case. And we could actually see that it worked also for fetal brains. And this is, I think, the first work that applies deep learning into the fetal FODs. And we also compare it with the classical methods. And an expert neuroanatomist, Professor Wazung in Harvard Medical School, preferred mostly this method. And yeah, in general. And yeah, so here, so basically what we did before is that we trained on newborns, preterm newborns, and then we tested on fetuses. But actually this problem of domain shift is very prominent in machine learning. It's also called distributional shifts in some other fields. And basically it's when your training set, let's say, is different or too much different from your evaluation or your testing set. And so in this case here, for developing brains, the problem is even more prominent. So for instance, we see that the brain changes very much at the beginning of life, as we've seen before in the first slide. And so yeah, so training basically a method on let's say a 30-week newborn might not necessarily, a preterm might not necessarily work for 40 weeks. And so since we are using MRI, the problem is even more complex. So there is also a shift in the imaging acquisition. So MRI has actually hundreds of parameters that you have to tune. So the protocols are so much different between different centers, and also within the same centers, the protocols can be, you can change basically so many, especially in diffusion MRI, that has the p-values, the number of gradient directions, etc. So yeah, so what we try here, and this is the work of a brilliant bachelor student, now a master student in PFL. So we basically try to use 2 data sets, the baby connectome project, so babies, I think from around like up to 5 years old, and then the newborns of the DHCP, so the data set that we used before. And then we try to see, okay, how does a method trained on one data set generalizes into the other. And also within, for example, the DHCP, we split into different age groups. And then we try to see, okay, how, like, we try to evaluate the shift, and also to somehow address it using either data harmonization methods. So in this case, we use the method of moments, and or domain adaptation. So basically fine-tuning on a small number of subjects. And actually what we found is that like fine-tuning with a small number of subjects can help a lot. And we also found that there is a higher shift in newborns compared to fetuses, but this is the paper if you want to look at it more. So this is just an image of the baby connectome project. So this is a crossing fiber, and we can see that using only 6 directions and the B0, we can actually get a crossing. And so people coming from a pure physics background might not necessarily accept or believe this result, but actually the thing is that the network learned a mapping. So it's not only coming from these 7 directions. So there is a lot of information in the training actually that is used. Yeah, so this is, so typically after computing FODs, you perform tractography. And so basically you try to map, to link all these FODs with a tractogram, and you get these kind of white matter fibers. And we can see that actually the results are also quite good with the deep learning method. There is also another work that I will not detail much here, but it's basically we try to use kind of a forgotten ground truth, the single fertility tissue CSD, in the context of developing brains and deep learning estimation of FODs. And this work was actually presented in Morocco by Rizong in CD MRI, MICAI. And yeah, the idea was basically we found actually that this ground truth is quite robust, so it improves much more with the number of directions, and also it's more robust to domain shift. Okay, so this was the first project. So the next project I want to talk about is basically the improvement of the true plane resolution with autoencoders in the developing human brain, so using neonates and fetuses again. So typically, basically you have the in-plane resolution, which is the XY here, and then you have the true plane resolution. And the idea is that in developing brains, so this true plane resolution is usually very high. So we have like thick slices of 4 or 5 millimeters, for instance, for fetuses. And it has been actually shown in adults that interpolation improves the different downstream tasks that you extract from the diffusion metrics. And but basically what we tried to do here is use deep learning to improve this true plane resolution. So yeah, so instead of basically doing the interpolation in plane, so we use an autoencoder. So this input is the same as the outputs, and then we try to learn this latent representation here. And we actually use this latent representation in the testing phase. So this work was actually inspired by cardiac MRI that also have the same problem, and their thick slices can even go to 7 or 10 millimeters. So in the testing phase, what we do is basically we encode 2 adjacent slices. So we actually remove the middle one as it's represented here, and then we try to predict it or estimate it using the encoding. So we encode these 2 adjacent slices, and then we get their encoding, and then we do a weighted average here in the latent space, and then we decode this weighted average, and then we hopefully get the encoding. We get the slice that should be actually in the middle. So this is what we've done, and we've done it actually so basically by let's say varying this weight, you can actually get many slices in the middle. So this is what we've done for either 1 or 2 slices. Yeah, so we use basically the developing human connectome project data to train and test, and also the fetal data from the Kinderspital of Zurich to test the method. So we actually got good results compared to traditional interpolations here. And these results are even better when we try to predict more slices in the middle. So for instance, here and here. So here we actually try to explore the latent space. And the idea was that, okay, if we have 2 images coming from orthogonal directions, so the gradient directions that we estimate the diffusion along are orthogonal, so they should be 2 different. So let's say the slice-wise Euclidean distance should be higher. And this is what we see here. So we have this when the angle is 90 degrees. So the distance is very high, and then the more you go towards 180, it's similar. It gets close. So we actually see the same pattern also in the latent space, and this was quite reassuring. So yeah, these 3 points, for instance, are mapped to these 3 points here. So basically from the diffusion weighted images, so the acquisitions, you can extract many metrics, and one of them is the color fractional anisotropy. That gives basically how different is the diffusion directions along in a specific voxel. And the color coding is that the main directions basically is color coded with red meaning left to right, and then green anterior posterior, and the right is superior inferior. And we also got quite smooth maps for this color FA. So we can see that, for example, here compared to this part, or even like here, for example, we can see that it has less artifacts, let's say, compared to here, this linear interpolation. This is the ground truth. But this can actually also be subject to over-smoothing, and when you over-smooth, usually you lose some details. So this can be like a disadvantage, probably. Then yeah, so again, with this apparent similarity, let's say, between preterms and fetuses, so we again trained the network only on preterms, and then we tested it on fetuses, and we could actually get smooth maps. So this is the original slice, this is the next slice, 5 millimeter later, and this is what's in between. And also in the subject that is even younger and that had motion. So we had to correct first for the motion, and then predict these 2 slices. But of course, a more thorough evaluation would be better. Okay, so now we're done with the diffusion MRI project. So now I will talk about this is actually my new interest. I'm getting more interested now in functional MRI, and my new lab is doing, like, it's an expert lab in functional MRI in PFL. And basically what I started to do with my previous lab, UNIL with Mel, is basically to parcellate the brain, parcellate the thalamus into its different nuclei using functional MRI. So this was actually the goal, and the thalamus is a very important structure in the brain. So it's it's in the middle of the brain, and it looks like a potato, and it's involved in both like the low level sensory processing or even cardiorespiratory tasks, but it's also involved in high level cognition. So it's an extremely important region of the brain, and subdividing it is very important. And it was done in the past. So I actually found 5 studies before which did it. So the results were somehow consistent, somehow not. So they used around between 20 and 100 subjects. They used this winner takes all. So basically the goal, if we see here, so we want basically every voxel to assign it to a cluster, so to a segmentation, let's say. So for instance, all these voxels here are assigned to this nuclei that is, let's say, primary motor, primary sensory, and then so this is the main goal. It's really to assign every voxel to a cluster, let's say. And basically, all of them use this winner takes all, meaning that they actually see so basically which voxel or which brain region connects most with this voxel, and then they actually neglect all the other regions in a way. And there are many concordant findings. So namely, there is this, for example, this salience prefrontal network that can be seen here or also here, and there is also there's the dominant somatosensory and sensory motor cluster also. And there is actually there is this default mode network that it's basically so when you wander, when you are not doing any task, so this default mode network actually switches on, and there was no significant connectivity to it. However, yeah, so there is a small number of subjects, so the winner takes all approach is quite coarse, especially for developing brains. So we want actually to use more information about the connectivity to other brain regions. And there was also no across-age development studies. So basically tracking the different nuclei across age. Okay, so this is actually what we tried to do. So here we are using a population-based study. So we basically have many subjects for different age groups. And then the idea is to really see across age how this clustering evolves. And the clustering is actually done with the k-means. So basically each voxel, let's consider each voxel as a vector. And this vector actually contains the functional connectivity to other brain regions, so the 40 brain regions. So the vector has 40 elements. And then we basically cluster all these voxels. And then, so we call it KNIT. So K means for nuclei in infant thalamus. And it's also based on some level of bootstrapping and uncertainty and confidence. So basically, we bootstrap on either the subjects. So basically, by taking randomly 75% of the subject of each age group, and then computing the functional connectivity, doing k-means, and then checking the overlap between the different clusterings or different segmentations. We also do the same with the time points. So this time we have basically 2,300 time points of the BOLT signal, so the blood oxygenated level dependent signal. And then we basically take only 75% of them. And then we see again, apply all the pipeline, and then we see the agreements basically, in the clusterings. So this is just a schematic presentation and also actually some results of the 42 weeks. So these are the different brain regions that are considered here. And this is an example of a 42 weeks subject. So first of all, all this clustering is actually done voxel-wise, but still we could get something symmetrical across the two hemispheres. And also we could get some physical proximity. So voxels that are assigned, so the clusters that are, or the voxels that are nearby are usually assigned to the same cluster, which is a good start at least. And so this is also the uncertainty map. So basically just taking the majority label across the different bootstraps on the subject's bootstraps. And so this is actually inverted here just for visualization, but we can see that the borders are more uncertain. So basically there are more black here, which makes sense actually, because these are the ones that you would expect to be more uncertain. So then we actually compared the method to the winner takes all approach. And for this, we had to basically merge the different labels. So basically these different labels are the different lobes. So we just took the labels that correspond to the temporal lobe, and then we assigned it to temporal, and then same for the parietal, the frontal, the occipital, and then the limbic areas, so the subcortical areas. And what we could get is actually the segmentation here that is less symmetrical, that has more these, it's basically symmetrical, and also we can see that it's actually not it's dominated by the frontal and the parietal lobe. And this is kind of a problem because the other information is actually lost. So this is basically where this k-means clustering here has kind of more granularity. So the vector space is higher. So probably this is why it performs better for this case. So what we see here is basically the functional connectivity of the oldest age group, so the 44 weeks, and how it connects to the different brain regions. So we can see some kind of patterns that happens across the different clusters. So for instance, this here, or this here. So there are some regions that are actually always highly, that the thalamus is always highly connected to, such as the parietal lobe or the frontal, or also this cingulate gyrus here, or the superior temporal gyrus. And so this cluster 3 is actually a noisy cluster that is very small, we will see later. So we basically discard it. And since actually this is a lot of information, so we basically merged the two hemispheres, so the right and left. Then we average them, and we also z-scored so that we can basically say, for instance, if it's higher than 1, then it's highly connected. If it's less than -1, it's more disconnected. So again, so this is the discarded cluster, this cluster 3. And we can see that these regions, parietal, frontal, superior temporal gyrus, are always connected in almost all the different clusters, but there is actually some specificity. So for instance, this cluster 1 has the occipital cortex, which is the visual cortex, basically highly connected. Or for instance, this cluster 2, which has the insula, and it has also the cingulate gyrus. So so basically this method that has some kind of high granularity, and it also uses these connectivities, let's say, to cluster the different voxels. So here is the evolution across age of the different nuclei. And we can see that this yellow one is basically a pulvinar, as in adult, so it's responsible for cognitive processing of the audiovisual sensory stimuli. And we can also see the salience here. That is mostly used for detecting and filtering the attended stimuli or the salient stimuli, so mostly some attention a bit. And then we also see this ventrolateral, so this cluster 5 here is here, and we also noticed actually this growing in volume of the dorsal cluster, and actually it's mostly connected to the frontoparietal network that is known for executive goal-oriented tasks. So probably the neonate starts to really use these cognitive tasks, let's say, maybe from here. This is just a hypothesis. And we also see actually that the symmetry increases with age. This is also another result. This is just the volumes, so, and how they vary with age. This is what we've seen. So basically the dorsal cluster and the ventrolateral going down, and the dorsal cluster going up. And here, so basically the idea is to see how let's say, are the clusters stable across age, and what are actually what is the number of clusters that we can use. So we can clearly see that actually the more we go with age, the less variability we have, so the more robustness actually. So we can actually parcellate the thalamus with more clusters the more we go with age, which makes sense in the sense of specialization. And this is actually the case also for the bootstraps on the subjects. But here, since the number of subjects is different, so this is a confounding. So we try to take it into account here. So it would probably, so this black curve actually would probably go down if the number of subjects didn't go down drastically. So if it was, for example, like this would probably go down. So so this is actually a solid result, I think, in terms of the robustness and the repeatability. And in the end, we actually choose 5 clusters as kind of a compromise. So we computed other metrics actually to decide on this, but it was mostly like a compromise between not a high number of clusters, and also so that we take the same number of clusters for different for different ages, so that we can compare them. So this is actually unrelated to the clustering of the thalamus, but it's just how much is the thalamus actually connected. So the whole thalamus is connected to the other brain regions, again, across age. And we can actually see that there is always these 3 regions that are basically the parietal, the superior temporal gyrus, that is actually responsible for the auditory processing, and the frontal lobe. Those are very high. And then we have some limbic areas that are low, and then we have some high slopes, for example, for the cerebellum. So since the thalamus is part of the brain, so we were saying, okay, maybe actually the connectivity of the thalamus is driven by the whole connectivity, or modulated by the whole connectivity with the brain. So this is what we try to see here by just computing the functional connectivity between the different regions of interest. And we could actually see some similarity, namely for this parietal and the frontal cortex that is very high. or even the cingulate cortex around here. And these matrices here are actually just differences of two consequent ages. So this is 40 weeks minus 39 weeks, and etc. And we could actually see almost the same pattern even in this direction, except for the superior temporal gyrus that is highly connected in the thalamus, but here it's extremely low. So it should be around here. Yeah, it should be around temporal gyrus. So it's not that high actually. So but I think it's ranked around the 19 connectivity, but in the thalamus it's actually ranked around like second, something like this. So this is probably very specific to the thalamus. Okay, so yeah, so to conclude, so we have found this increase in robustness across age. We have seen also this increase in connectivity at the age of 41 weeks. And also that this connectivity might be actually driven by the whole brain connectivity, except for the supratemporal gyrus. We have seen, actually it's in more details can be found in the paper, but that this K-means actually it's probably better than this winner takes all. And yeah, these are the four nuclei that we found. And actually, since I'm getting interested in consciousness, so I tried actually to see if we can somehow link this work or try to see the literature about consciousness. And I found that the thalamus is very involved in consciousness. Hence, we can, for example, make some hypothesis linked to the salience network, for example, and if its beginning actually maybe means like kind of a self-referential consciousness that is starting, or even when we see, for example, this burst at 41 weeks in the connectivity. So I will present actually this work next week in NYU in the Infant Consciousness Conference, and yeah, discuss basically these ideas with more expert people on this. Yeah, so with this, I would like to thank all my coworkers, my collaborators, the Swiss National Science Foundation for the funding, and the Developing Human Connectome project for making their dataset publicly available, and you for your attention.
Thank you very much, dear Hamza. Very nice presentation and very impressive. I liked how you showed like three projects at once in a very nice and simplistic and also concise way. So yeah, congrats again for all the work you are doing. Now time for questions. So we did receive many questions, but I'll try to select some of them and also probably give you the questions by like team or by class of questions. Let me start, for example, by these like direct questions. So the first one is probably regarding the first project you talked about. So the question says, yeah, why do you use aspherical harmonics decomposition? And you are muted, dear Hamza.
Yeah, sorry. So this is actually this is kind of the standard way to do in diffusion MRI, because it's a signal on a sphere. So, and this spherical harmonic decomposition really is the best mathematical representation that you can use later to basically manipulate these mathematical objects. So this is very much detailed in the thesis of Maxime Descoteaux. So it's written Descoteaux in 2008. And yeah, so basically the idea is this. The answer is that it's a spherical signal, so you have different gradient directions, and yeah, this is the best way to basic. And also there is maybe another reason that we exploited here. So when you project into this spherical harmonics basis, you are somehow, you make your input independent of the acquisition, in the sense that if you have different acquisitions, let's say with different gradient directions, so the spherical harmonic somehow normalizes this. So these are maybe the two reasons.
Okay, thank you. Next question is about the unit you used in your first project. So again, a direct question, is this a 3D unit and what was the loss function used?
Yeah, so it's a 3D unit. So we basically used different. So basically, we just patches of 3D shape. I can show it here. Yeah, of 3D shape, and we just used the mean square error, so it was working quite well. And yeah, the idea is basically, so the main goal here is really not like to innovate, let's say, on the architecture, but still we aim to add these kind of extensive residual blocks to, so because the information is very it is very low, let's say, so it's not, we are only using 6 directions. That we want to predict is very high dimensional. So we try to basically use these residual blocks so that the information flows maybe better. And also, actually, one of the main points why this method works is also because we take the large patches. So this is 16 by 16 by 16. We tried different ones, but basically we have a lot of information. So, and there is actually some neighboring information between the different voxels. So yeah, so basically the network uses this information.
Okay, yeah, there was another question which is related, I mean, on the architecture, but I think you already like answered. So the goal here was not like to have the best architecture, but so the question, but probably you'll be interested. The question was on using attention in the unit. Have you tried like a version with attention layers? Uh, yeah,
No, so
yeah.
I didn't, but that would be maybe something
Yeah.
interesting to try first.
Okay, thank you. I do have one question which is not really machine learning related, but I'll give it to you, dear Hamza. So what is the software you use for visualizing the FODs?
So this is a matrix here. It's called a matrix, and it's yeah, for the processing we use both DiPy and a matrix for the processing of all these FODs, and I mean encapsulated in Python. And to visualize FODs, so it's a MR view, which is part of a matrix basically.
Okay, I'm not very familiar, but just it's my question. Are these like open tools or proprietary tools? Okay, they are open tools.
Yeah, they're
Okay.
open
Okay.
access. Yeah, it can be downloaded. It's free. It's perfect.
Okay. Thank you. Next question is quite interesting. So, did you collaborate with medical doctors in this project, and how was your experience?
Yes, it's a good question actually. It's a question that always also comes in this environment, like how to optimize, let's say, work between doctors and scientists, radiologists mainly here. So yes, yeah, many of these works actually here they work for. So for example, for evaluate, so one of the areas in which doctors are very helpful for us, because we are not knowledgeable in that sense, is basically when it's about analyzing the basically these FODs, for example, or the white matter bundles. So they have a lot of neuroanatomical background, so they know how let's say the configuration of the brain, how the cortex should be at that time, how the white matter, how the cortical place and everything should be at that time. So they can basically use this knowledge to assess our estimations of deep learning. And yeah, typically when we don't have a ground truth, so for example, for the fetal brain, so because we cannot acquire long acquisition, so we don't have a lot of directions, so we cannot have a ground truth. So they are typically very helpful there, and yeah, this is.
Okay. Okay, thank you. Next question is probably maybe on the second. Yeah, yeah. Let's now move to the second project you presented. So first question is the domain shift problem you described is interesting. How did you deal with it? Very general question.
It's a very good question. I think there is no solution so far. So yeah, even in, let's say, LLMs, for example, if you have a model that is trained on classical Arabic and then you want to use it in Darija, you have to somehow fine tune it or something like that. So this is the typical problem here, and we basically dealt with it with two methods. So there are two methods that were used here. So I think namely the domain shift problem can be tackled from 2 points. So either like trying to work on the data and let's say remove what should be removed so that the data sets, let's say, don't have the component, or like the component of scanning and of acquisition is somehow reduced between them. And or you can work on the model. And in the model, so here we just use actually fine tuning. So we really just
Okay.
train on some subjects and then we fine tune on other subjects. And for this one, we use the method of moments, basically.
Okay, thank you. Second question is also an interesting one I find. So, in the second problem, why not using a VAE instead of an autoencoder?
That's a very good question. So I actually started working with the VAE in the beginning for like 1 or 2 months, and then the results were not quite good. I'm not sure why. Probably because there are two losses that are fighting each other.
Yeah, yeah,
And actually, just so once I actually removed one the loss that makes the latent space more structured and Gaussian
yeah.
and everything.
The
And
KL
then
divergence.
exactly, and then it worked quite well. So maybe it's not needed. Maybe that's my understanding.
Very interesting. Yeah, very interesting problem indeed. Okay, yeah, for the person who asked the question, probably in the breakout session, you can have more details. So I do, yeah, probably I will take just two last questions to not take a lot of time. The first one is quite, it's, well, it's funny, but yeah, interesting. How was Mikai in Marrakesh?
I think it was good. It was really good. I mean, the organization was good. I mean, I didn't notice anything very bad.
You,
So it
you
was
were
quite
there? You were there, Hamza, or?
yes, yeah, yeah, I was
Okay,
there
you
actually
were
with my
there.
lab.
Great,
So I
great.
was doing the local guide and at the same time trying to
Nice,
attend the talks. Yeah,
nice.
it was good. I think people were happy. I mean,
Great, nice to hear. Yeah, the other question, which is quite interesting too. So, how do you measure the generalization or generalizability of your method if we want, let's say, use it in Morocco?
in Morocco. Okay, it's a
Mm,
very vague question. So in Morocco, you would probably so maybe the person who asks this question thinks about I don't know different like training on one ethnicity and testing on another.
yeah, probably.
Uh,
Probably.
my answer would be I'm not sure. Probably yeah, you need to have a very large data set and very mixed and for sure, I mean these data sets. So the data set actually that we use here, they come usually from London. So
Mhm,
I'm
mhm.
not sure actually about the diversity in the ethnicity, for instance. Or there is the Baby Connectome project. I think it comes from US. So yeah, so I mean,
Okay.
yeah, this is a good question, and we probably need to gather data and also make it maybe publicly available if possible. And this thing.
Okay. Thank you. Yeah, let's take a last question and then we move to the breakout session. So, can you give us an idea on the compute cost to train the 3D units?
Yes, I was using a computer in Boston which had an A6000 NVIDIA GPU, and it was taking I think around 40 hours per model, something like this.
Okay.
So it's quite heavy. It's
Mm, mm.
quite heavy, but yeah, but then when it's trained, it's good. I mean, testing is just 1 minute per subject or something like this. So it's yeah.
Thank you. Thanks a lot, dear Hamza, for the nice presentation and also for the exchange. Thanks to the participant for their questions. Very interesting questions as usual. And yeah, now, well, time is running. Sorry for the other questions I didn't took. Now time to go to the breakout session where you can continue discussing with Hamza. We will have two breakout sessions as usual. One called Ask Me Anything with our speaker, Dr. Hamza Kebiri, and the second breakout session will be just like a very general one for networking. So now I will take just yeah, and thank you again, Hamza. It was a
Thank you very
pleasure
much.
having you with us
So,
in MoroccoAI webinars. So yeah, I'll take a little moment to stop the recording and then we'll move to the breakout session.